This is part 2 of the chapter 15 lecture. So in part 1, we discussed B cells, which are part of the third line of defense, acquired immunity. The second cell type that's important for third line of defense are the T cells. So we have two types of T cells, which we've mentioned briefly in the last lecture set. The cytotoxic T lymphocyte is a T cell type that has MHC1 on its surface because it's a human cell. It has MHC2 on its surface, and it also will have CD8 on its surface, which is an identifier of it as a cytotoxic T lymphocyte. The role of the cytotoxic T lymphocyte in acquired immunity is to recognize and respond to endogenous antigens, which are antigens that get inside the host and then further get inside of a host cell. The other type of T cells that we're focusing on in this course are helper T lymphocytes, which will express MHC1 on their surface as a self-human cell, MHC2 on their surface, and will also express CD4 receptor on their surface. The helper T lymphocytes will be responsible for dealing with exogenous antigens. And those are the antigens that get into the host but do not infect a host cell, so they're freely floating inside the host. So the helper T cell will respond to antigens of the first category, and the cytotoxic T cell will respond to antigens of the second category, and neither should respond to antigens of the third category. So one thing about T cells that we touched on briefly in the last lecture set that makes them very different from B cells or other antigen-presenting cells is that T cells have to be presented with an antigen on MHC molecules. So we have the antigen-presenting cell will first encounter the antigen, which in this case is the pink bacillus bacteria, brings the antigen in, processes it, and puts part of the antigen on its surface. Because this was an exogenous antigen, it will be presented on MHC2. That is all essential and has to be done before the T cell can recognize the antigen. So T cells only recognize antigens that have been processed and presented on MHC1 or 2. So in the second box below, we have the antigen presenting cell in purple, which is presenting this exogenous antigen on MHC2 molecule. Because the antigen is being presented, the T cell can now recognize it. And the T cell that will recognize exogenous antigens presented on MHC2 is the helper T cell. So we see the helper T cell bind to the presented piece of the antigen with their T cell receptor, and after this, the T helper cell will orchestrate or organize a response of multiple types of immune cells. So we see that response from the T helper cell in this figure. So initially in this figure, we have the antigen presenting cell, which has encountered an exogenous antigen, processed it, and presented it on MHC type 2. The CD4 helper T cell recognizes the MHC2 presented antigen with its T cell receptor, and then will allow the helper T cell to activate itself and other immune cells. So the CD4 helper T cell, when it binds to a presented antigen, can either become a memory cell, which will have a long lifespan, will stay circulating in the host if the antigen returns at a later time, or will become a T helper cell, which is divided up into 3 groups in this figure: TH1, TH2, and TH17. All of these T helper cells we're just going to lump together into T helper. So T helpers are going to organize inflammation and recruit other molecules to the area, including phagocytes, to destroy the antigen. Some T helper cells will also activate B cells to become either regulatory plasma cells and make antibodies, or memory B cells. And then we also have the possibility that the T cell will be activated to become a T regulatory cell, or T reg. And this will just make sure the immune response doesn't get out of control. The cytotoxic T cell, which expresses CD8 on its surface, also has to have antigen presented in order to recognize it. In this figure, we are dealing with an endogenous antigen. So CD4 helper T cells respond to exogenous antigen. CD8 cytotoxic T cells respond to endogenous antigen. So in this case, we have an antigen that has invaded the host and also has invaded a host cell. So the cell that is infected with the endogenous antigen will process the antigen and we'll present it on MHC type 1, as this antigen presenting cell is doing. When the endogenous antigen is presented on MHC type 1, the cytotoxic T cell will recognize it and will tell the infected cell, the infected human cell, to destroy itself by apoptosis. And the way that's organized is the cytotoxic T cell will produce molecules called perforins and granzymes, and those will cause the infected host cell to undergo cell suicide, also called apoptosis, and be destroyed. The reason the cytotoxic T cell causes a human host cell to be destroyed is that it's the only way to kill the endogenous antigen that is inside it. So this figure summarizes all of the T and B cell responses of the adaptive immune system. So we started off in the top of the figure showing that the lymphocytes are a type of white blood cells. They start as stem cells and they either develop in the bone marrow into B cells or in the thymus into T cells. That they then will respond specifically to antigens if they can bind to them with their B or T cell receptor. So we have on the left side of the figure the B cell response, where we see an exogenous antigen make contact with an antigen presenting cell. It will then be incorporated into the antigen presenting cell, processed and presented to a T cell, which can then become an activated T cell, a memory T cell, or a helper T cell that will activate B cells to then make antibody as plasma cells, B memory cells, or regulatory cells. Those activated T and B cells and antibodies will then circulate in the host and try to bind to more of the antigen and destroy it. Not shown in this figure is the cytotoxic T cell response, where the infected human cell would simply undergo apoptosis and be destroyed by a signal from the cytotoxic T cell. Okay, let's shift gears a little now and go into applied immunity. So applied immunity is the concept of using the immune system to protect us against potential future infection. So we've mentioned that B and T cells both can become memory B or T cells and remain in the host in case the pathogen returns at a later time. So this figure shows over time on the x-axis and antibody titer, which means the concentration of antibody in the blood circulating through the host, on the y-axis. So we have the first exposure to antigen, which is abbreviated AG, at the very start of the x-axis at time 0. You see a period of latency where there's no antibody production, and this is because the second line of defense has responded. It was not successful in clearing the pathogen, so now eventually the third line of defense becomes activated and starts producing antibody. So we see the red line representing IgM, which is the first type of antibody made in response to a pathogen. We have the light blue line representing IgG, which will take over eventually as the infection is eventually cleared, and the total amount of antibody in purple. So we have the primary response, which after a period of latency where the response is getting ramped up, responds to the pathogen, and the pathogen is cleared and the host returns to normal pre-disease state. If the host is exposed to the antigen a second time, we have a 0 latent period, so that is absent, an almost immediate response of IgG production and some response with IgM production. The response is also much bigger this time, and so the secondary response is how the immune system reacts to the second exposure with the same antigen. The reason the secondary response is so much faster and bigger is because the host already has memory B and T cells from the primary response. So we use these concepts, and there's different ways that a host becomes immune to an antigen. So after a host has been exposed a first time and develops memory B and T cells to that antigen, that host is considered immune to that antigen. If the immunity was a result of exposure to the antigen in everyday life, this is natural immunity. If the immunity is the result of exposure to the antigen at a healthcare office, this is artificial immunity. So natural immunity and artificial immunity can be active or passive. So naturally acquired active immunity is when the person is exposed in daily life to an antigen, their immune system responds by making antibodies. Naturally acquired passive immunity is when the individual is exposed to antigen in everyday life, but they are acquiring immunity by being given antibodies. So an example of naturally acquired passive immunity is breastfeeding, where the mother gives immunity to the newborn by giving antibody in the breast milk. Artificially acquired active immunity is when you go to a healthcare office and you are exposed to antigen, so your body responds by making antibody. Artificially acquired passive immunity is when you go to a healthcare office and you are given antibody directly. And those different categories are defined in text here, if that makes more sense to you. So with active immunization, we have this illustrated with the red line. So we have antibody concentration on the y-axis in this graph and time on the x-axis. The initial inoculation is the initial exposure to antigen. Because the exposure is to antigen, it is an active immunization. So you are exposed to antigen, your body actively responds by making antibody. With each of the boosters, we have additional exposure, so we're ramping up the memory response, and the memory cells are responding more and more robustly. So you get a very high concentration of antibody that will last a long time with the red line. In contrast with passive immunotherapy, this would be going to a healthcare office, and the vaccine you get injected with contains antibody. So you have a lot of antibody initially after the injection, but you eventually over time the antibodies degrade, so you lose immunity with passive immunotherapy. So active immunization, like exposing the individual to antigen as opposed to exposing them to antibody, is preferable because it confers long-term immunity to an antigen. So there are several types of vaccines that we can use to give people a primary exposure to antigen, which they then will be ready, if they're really exposed to antigen, with memory B and T cells and have some immunity to the antigen, so hopefully will not get sick, or will get sick in a much milder way. So one type of vaccine is the whole cell vaccine. So these can be either killed or inactivated microbes, or they can be like microbes that are alive, but they are attenuated, which means weakened. So in a whole cell vaccine where the microbe is killed or inactivated, the microbe is exposed to heat or chemicals so that the microbe is dead, but it's still antigenic. So it will still stimulate production of antibodies, which will result in memory B and T cells and the immune response will be active. If the microbe is alive but has been attenuated, it's attenuated again with exposure to chemicals. So it will be alive, but it will not be pathogenic or able to cause disease. It will still be antigenic, so will still induce an immune response in the host. Subunit vaccines are when a part of the microbe is taken and injected into the human, and this part of the microbe will not make the human sick, so they are not pathogenic parts of the microbe. They are antigenic, so they will induce an immune response and production of antibodies and memory B and T cells. However, they will not make the individual sick. So we have some examples of viruses where you could take one of the virus surface proteins and inject that as an antigen that will confer immunity but not make the individual sick. We could take molecules from a bacteria. In diseases where toxins cause damage, you can inject a modified form of the toxin called a toxoid. Or you can do genetic engineering where you artificially create a protein that would be present in the microbe. You can inject that protein, so you'll then get an antigenic immune response, but you will not make the individual sick. So here we have an example of a subunit vaccine. So this is the vaccine against tetanus. So tetanus is a disease caused by bacteria, but the damage in tetanus is from the tetanus toxin. So by altering the toxin into a toxoid and injecting that into humans, you can initiate a primary immune response, which will not make the individual sick, but will give them antibody and memory cell protection against the tetanus toxin if they're ever exposed to it. So with part A, we have a artificially acquired active immune response. And so we have the lag period or period of latency. We have production of IgM antibody first and then IgG antibody second. And after that, immune response settles down. In the person's actual life, they are exposed to the tetanus toxin. So because they've already seen the toxoid, their response to the toxin will be their secondary response, which will have little or no latent or lag period, and the production of antibodies will be much bigger thanks to the memory B and T cells. This type of immunity will also be long-term because it is active immunity. There is still a lot of people that are part of the anti-vax movement, which believe that the risk of vaccination exceeds the benefit. A lot of this is based on a paper that was published in 1998 that linked the measles, mumps, and rubella vaccine to autism. 2 years later, that paper was retracted by the journal. The findings were disputed by several researchers and have been disputed by dozens and dozens of papers since, but it still held the public's attention. We currently have a mumps outbreak in the United States as a result of low vaccination rates, and there's a lot of celebrity and numbers in the anti-vax movement, which is very dangerous for public health. Vaccinations are crucially important for establishment of something called herd immunity. So this is the idea that if a critical number of people in the population are vaccinated and protected against a disease, diseases cannot spread through the population easily and cannot spread to vulnerable individuals in the population like young children and very old adults. So in the top image, we have a population where most of the people in the population are not immunized, so they're mostly blue. We have a couple of people that get sick with a contagious infectious disease. And because we don't have herd immunity in this population, the disease spreads rapidly and we have an epidemic or potentially a pandemic. In the middle scenario, we have a population where most of the people are not immunized. There's a small number that are immunized, and there's a couple people that get sick. In this population, because we have not established herd immunity, the disease will spread very quickly through the population. In the last scenario, we have a population where we've established herd immunity, Most individuals in the population are immunized and protected against the disease. So when a few individuals that are not immunized get sick with a contagious infection, the infection cannot spread through the population because of protection by vaccination. So vaccinations are critically important for public health. Their benefits far outweigh their risks, which are very rare and not a reason not to get vaccinated. That concludes chapter 15.